Assalamualaikum and hello everyone, here I'm back. I'm Siti Nurhana binti Maknur with metric number of 207193. Today, I would like to show how the rate of alcoholic fermentation in yeast is measured with different types of sugar. Before I proceed, let me give a brief introduction on that. Alcoholic fermentation is a biological process which converts sugars such as glucose, fructose, and sucrose into cellular energy, producing ethanol and carbon dioxide as the byproducts. It occurs in yeast, bacteria, and other microorganisms as well as oxygen-starved muscle cells. Because yeast perform this conversion in absence of oxygen, alcoholic fermentation is considered as an anaerobic process. Alcohol fermentation has many uses, including alcoholic beverage and ethanol fuel production, and also bread, those rice. So, for this experiment, I'm gonna do an alcoholic fermentation in yeast to measure the rate of yeast fermentation with different types of sugar, which are glucose, table sugar, and starch. For the observation, I'm going to observe the production of carbon dioxide by capturing it in a balloon and measuring how much the balloon inflates over a period of time. Let's explore it!
Firstly, you have to prepare 4 empty plastic bottles. Label all the empty plastic bottles with glucose, table sugar, cornstarch, and control. Here I'm using a sticky note for labeling. After that, fill in each of the empty plastic bottle with 70 ml of warm water. By using the filter funnel, add 21 teaspoons of carbohydrate samples, which are table sugar, glucose, and cornstarch, into the labeled water-containing bottles respectively. For the control sample, there's no carbohydrate is added into this bottle. There will be the mixture of only water and yeast, and it acts as a control for this experiment. Then I'm using Mauripan instant yeast for the source of the yeast. Add 3 spatula of this dry yeast into each bottle. Mix them by putting thumb over the top of the bottles and gentle shaking. Then, the opening of each bottle is covered with a balloon to capture the gas produced during this experiment. After this, all the bottles are observed every 5 minutes for 25 minutes on what occurs in the bottles and any changes in the balloons. And then, let's start our observation. So here is the results and the size balloon observed.
So, have you guys seen the result just now? It's really accelerating, right? Okay, here is the principle. Yeast is the fungus and needs a supply of energy for its living and growth. Yeast can use oxygen to release the energy from sugar in the process called as respiration. However, if oxygen is short, the yeast can still release energy from sugar. From this experiment, when it is mixed with sugar, the sugar has supplied this energy. The yeast eat the sugar and they turn it into energy and produce carbon dioxide through fermentation. The balloons on the bottles with yeast and sugar capture carbon dioxide produced by the yeast. It is this carbon dioxide gas which makes the bubbles in the bottle, causing the balloon to rise. The faster the rate of fermentation, so the faster the balloon will inflate. The balloons expand as the gas from the yeast fermentation rises. Different types of sugar give different effects to the balloon to inflate. In the bottle that contained yeast without sugar, the yeast did not have food, so the balloon was not inflated. As yeast does not have an enzyme to access sucrose energy, the table sugar, which is a sucrose, does not perform as well as glucose, which is a simple sugar. Fructose, galactose, and lactose also produce very little, if any, respiration in yeast occurs. So that's all from me. Thank you.